Hi everyone, I am assigned with the topic of monoclonal antibodies. Let's get started. Introduction. Monoclonal antibodies derived from a single clone of plasma cells that all having the same antigen specificity and produced against a single epitope of an antigen, while polyclonal antibodies arise from multiple B cells clones. A mixture of antibodies that detect different epitopes of an antigen, and the polyclonal antibodies are typical in a serum response to antigen. Monoclonal antibodies are the antibodies from a single parent clone. Antibody from one clone to detect one epitope of an antigen, usually produced in a laboratory using hybridoma technology. Next, let's look at the principle of the monoclonal antibody production. The principle is known as hybridoma technology. Hybridoma technology is nothing but the clone of a B cells stimulated against a single epitope of an antigen is fused with an immortal cells, thus the myeloma cells, which is capable of multiplying indefinitely. To produce a hybridoma cell, this has two unique properties include the first one is to produce monoclonal antibodies of same antigen specificity due to the B cell component, and the second one is multiplies indefinitely producing clone of the identical cells due to myeloma cell component. Next is the procedure for the production of monoclonal antibodies. The mouse is immunized by specific antigen against which monoclonal antibodies have to be produced. Next one is the after 22 hours of immunization, spleen is collected from mouse antibody producing B cells. Next step is the B cells are fused with immortalized myeloma cells by polyethylene glycol or Sendai virus. The fused cells are incubated in the het medium. Myeloma cells are nothing but used as a source of immortal cells. Myeloma cells are cancerous plasma cells, closely resembles mouse B cells, hence are compatible for fusion, have the capacity to produce their own antibodies. Fusion using PEG broth include fusion, the mouse splenic B cells and mutated myeloma cells are fused in PEG broth. In the reaction chamber, three types of cells will be generated. The first one is unfused myeloma cells, and the second one is unfused mouse splenic B cells. The third one is the fused hybridoma cells that we used in the production of monoclonal antibodies. This is the diagram showing the steps involved in the production of monoclonal antibodies. The isolation include by subculture on hat media. The next step is to remove the unwanted cells and to propagate the clone to hybridoma cells. This is carried by subculturing the cells in reaction chamber onto a special medium called hat medium. Hat medium contains hypoxanthine, aminopterin and thymidine. Purine synthesis in mammalian cells. Thus, splenic B cells acquired by either de novo or salvage pathways. Aminopterin blocks the de novo pathways so that the cell has to perform the salvage pathway to synthesize purines for its survival. Salvage pathway requires two important enzymes, HGPRT, thus the hypoxanthine guanine phosphoribosyl transferase and the thymidine kinase. So any cells of myeloma such as myeloma cells that lacks HGPRT can grow on hat medium. Fate of cells in the hat media include the unfused splenic B cells can grow but do not survive long as they are not immortal. The unfused myeloma cells can grow as they lack of HGPRT enzyme to perform the salvage pathway of purine synthesis. Third one is the hybridoma cells. They can grow and survive long. The selection of individual hybridoma cells include many B cells would fuse with myeloma cells to produce a mixture of hybridoma cells, each having specificity for one epitope. Hybridoma cells producing the desired monoclonal antibodies are selected by radioimmunoassay or ELISA technique. The application of monoclonal antibody production includes treatment of various inflammatory and allergic diseases and cancers. Monoclonal antibody can be used to detect pregnancy only 40 days after copulation and in the rapid diagnosis of hepatitis, influenza, herpes and streptococcal diseases. This is the diagram showing the production of monoclonal antibodies. That's all. Thank you.